Well, thank you all for coming this morning, and I invite you all to come visit me on West 16th Street, where the undergraduate fine arts department resides. We have 54,000 square feet. We have every new technology you can think of, and we'd like to see graduate students come by and perhaps even audit our classes. When I actually proposed to our president, David Rhodes, the inauguration of a Bioart laboratory, he had no idea what I was talking about. And most people don't know what bioart is and why it's not science. We're going to sort of talk about that here today. But in more recent invocations of bioart, David Rhodes is very proud of the fact that we are the first institution to have a bioart laboratory as part of a fine arts program. Next year, the Chicago Art Institute is going to set up their own. So this is the beginning of a trend, I would say, that bioart is part of as an international idea because we live in a biological age. The biological age is sort of the umbrella that affects the clothes we wear, the food we eat, the pharmaceuticals we take, the surveillance we're under, et cetera, et cetera, et cetera. And there is no escaping this kind of idea. Biology is technology now. And rather than the zeros and ones, which is a binary system, biology can offer twice as much coding effect by using the 4 nucleotide bases. But the question here today is, where is the art in bioart? Why is bioart not science? And let's take a look at some of this. I've written the definition of bioart for the Oxford definition of art, the Oxford Dictionary, of art. Many of my compatriots think that bioart is only related to wet lab practices, but that is an insufficient definition. For me, bioart is divided into 3 subgenres, one of which is the wet lab, as we see in Mark Quinn's portrait of Sir John Sulston, a major scientist in the UK who had given Mark some of his cells, and Mark's portrait of Sir John Sulston has been acquired by the National Portrait Gallery in the UK. It's sort of a very abstract notion of what the body looks like based on cellular information. And even in Paris just now, the finger of the dead terrorist, through DNA analysis, was able to determine who that was. So this question of identity, in part, is wrapped up in the code that exists within us. We carry with us a bioarchive of all of our ancestors, etc. Next is Patricia Piccinini, an artist from Australia, and this is a piece of hers called The Young Family from 2003, in which she creates this hybrid sculpture that is made of silicone but looks like it is a cross between a human and an animal, raising the kinds of questions that that this new technology poses, such as how human does an entity need to be in order to have the rights and privileges of human beings. And this is a question that chimpanzees and gorillas and monkeys sort of have come up in this discussion, but now we can cross between species in ways that were never possible before. So that is a kind of iconic image made in traditional materials that represent what bioart is. And the third subgenre is the way in which new technologies such as rapid prototype sculpture can add to this discussion, in which zeros and ones can create form. Form can be decoded in this way, and new kinds of imaging devices are available, such as one could see in remote sensing here, which was taken from the data garnered from a photograph. Bioart's been around since the late 80s. Tony Craig's Code Noah talks about the commodification of DNA. You don't own your own DNA, so this is also very anti-intuitive. One would think that because it's in your body, it belongs to you. It does not. And he uses the teddy bear as a kind of commodification icon to begin to talk about the work of life in the age of mechanical reproduction. Dennis Ashbaugh uses this kind of electrophoresis gel, which we became known when O.J. Simpson trial was going on, as a way to open up color field painting. Here are a few of my own artworks. Semiotics, which uses the chromosomes of many animals to depict the idea of sequence. And by looking at the kind of chromosomes and the size of the chromosomes, one can tell whether we're looking at an elephant or a fish or monkey or a flower. This was at the Getty Museum in 2001. Several other pieces, these series of silkscreen prints and paintings, was a way to talk about the chromosome as an icon in an illuminated manuscript. These are essentially silkscreen prints on frosted mylar, but added to the mix of chromosomes, one can see these double bodies, which are taken from Canadian Air Force exercises, because the word chromosome in Greek means colored body. So whoever looked through and saw these entities was able to discern in an anthropomorphic sense the nature of the colored body. So essentially, what we have here with chromosomes is the way the body writes itself. So the chromosome is an alphabetical form that is used to write the body. Another recent piece here from 2009, Astroculture, shown at Exit Art, in which I used LED lights to grow vegetables in the gallery. And in this piece, one can talk about the way in which agriculture is different when it is being performed in outer space, which is what is happening now. And here you can see a current piece, which is up now till April, and this is at the Cathedral of St. Divine, in which fuchsia is the new green. And you can kind of see in this series of photographs of the pieces that liquid light almost defines the visual image. Another artist I think who's very important in this discussion is Heather Dewey Hagborg, who is opening a biolab at Chicago Art Institute. What she did was to pick up gum and cigarette butts in Brooklyn, take them to Gen Space, and look at the information that was left in the DNA. She then did a series of scans of recognition of the facial recognition programs, and was able to tell many things about the person who left this debris behind, such as skin color, lip size, nose size, eye color, etc. She could not tell their age from these, this is called SNPs, and she had created a number of these portraits using rapid prototype sculpture. Natalie Girard thinks that there's not enough green space in New York. I agree with her. So she proposes to use the space for fire hydrants as kind of little tiny parks that would exist all throughout New York. And the worst thing that could happen here is a few plants would get trampled on. Mark Dion, another New York artist who works with archaeological techniques, had a residency at the Natural History Museum in London for the 300th anniversary of Linnaeus, who's the father of taxonomy. And when he got this residency and began his installation, the scientists there were very offended. They thought that putting an artist in a scientific institution was a form of degrading the importance of science. So part of his installation, which was very interesting, was to put fly paper on the top of this car, have his friends have butterfly nets out the window, and he would drive as fast as he could during a big avenue in London collecting insects. And when they were decoded for their DNA, he discovered 2 new insects, never before seen in London. So that was the end of the scientists thinking that artists have nothing to contribute, because clearly this is not the scientific method. This is only an artist would do something like this. Another set of artists, Heather Aykroyd and Dan Harvey, also from London, used grass as their matrix for photographs. And this too happened by accident. They were sort of at home one day and they forgot to put their ladder away. And when they finally put their ladder away, they saw that the color of the grass underneath the ladder was different than the other grass surrounding it. So they too worked with a number of scientists developing photosynthetic varieties of grass. And you can see at the scale, this is really impressive. So it is a living photograph, which sounds like an oxymoron. Another artist, Jalila Assadi from Amsterdam, worked with a number of scientists there and a number of scientists here in Utah to try to create bulletproof skin. The idea was to work with silkworms who can spin a kind of entity that is harder than steel. And this experiment almost worked. Didn't quite work, but that's not the point. The point is, when we think about speculation and we think about the future, we sort of inch up towards something like this. Christina Agapakis and Sisal Tolas created this project. They wanted to know why some cheeses were so stinky. And they began to take samples from the bodies of many of the friends on the team. So, for example, we have bodies of a philosopher's toes, somebody's nose, an armpit, etc., etc. And in culturing this cheese, which they did not eat, but they essentially found comparisons between the kinds of bacteria that are on people's bodies and the cheese that we do eat. So they were able to make comparisons that way without eating the cheese. In the Biolab here at SVA, we do painting with bacteria. We use an E. coli bacteria. It's not the same one in your gut, so don't worry about that. And we are able to transform plasmids as a painting material. Here you can see the New York City subway map in E. coli. Most recently, there was a young artist, who I don't know if he was young, but it was an artist, who went around the subway lines to test the subway poles on each one of the lines. And he found Streptococcus, he found E. coli, he found Salmonella, he found feces, he found a number of things. So that may be something of interest to you as you kind of proceed in your transportation needs. Yaakov Avrahami, who is an Israeli artist, used the E. coli as a form of watercolor. Leora Euclea decided that there was too much wood being used for furniture, and she thought that she could substitute the wood using microform, which is a kind of mushroom that is replacing styrofoam. And this is sort of what it looks like when you grow it. It's quite beautiful. And here is her table that is half IKEA and half mushroom. And finally, these are some of my pieces. These are rapid prototype sculptures called remote sensing, and they're based on the kind of satellite data that is used in order to visit places that are either too toxic or war-torn to kind of assess the landscape. And these come from these little Petri dishes, and these are vanitas in a Petri dish, bringing the 18th and 19th century idea of vanitas, which is the fact that everyone's going to die, into focus. But in these cases, there's a combination of animal, vegetable, and mineral, which is what is happening in synthetic biology. And here you can see the extrapolation of the 2D to the 3D. I had no idea what was going to happen when I did this, but I did it, and I was very pleased with the results. And just in conclusion, we were the first school to enter a synthetic biology competition called iJam, and we entered in the art and design track. And what we did is we put an email out asking people in our community whether they wanted their soil tested for nutrients. And our project was called Soiled, and essentially we tested 60 different sites and learned all about sort of the ethnic orientation and the personal stories as well that people had with regarding their homes. Now, you may think growing a tomato in your backyard is good for you, and it may be, but it may not be. Okay, so we did this chemical analysis of all of the nutrients in soil, which was based on color as a metric. And what we were able to find is the variety of different nutrients are very different from plot to plot. And we designed this microfluid device which can use these soil samples as a way to eliminate the toxicity of the soil sampling device. And here we are using processes involved in art to create this discovery. And our project, which was based on LED lights lighting up for each burrow, based on the amount of nutrient in the soil. And here is a kind of photograph of it. And this was our installation in Boston. And this is our award, okay? We got a gold medal for the best art and design project. We're also nominated out of 300 teams for best integrated human practices, best education and public engagement, and best presentation. So we're very excited. Thank you.